So it seems that very few students have joined my Google Classroom. I will not post any material in one WhatsApp group. So if you need, you have to join the Google Classroom. And from next day onwards, next class onwards, I will not post link on WhatsApp also. So those who are not on in my Google Classroom, be very clear, you are not in my class also. Okay, to start today's topic, we will pick up after we had studied the Watson-Crick DNA model. Today we will go on the different types of DNA model helix. So let us talk about what is A DNA, B DNA, C DNA, all the Z DNA helices. So we had discussed about structures, and I have already posted the notes on this. So now we will talk about what are the different types of DNA. So we know that Watson and Crick model, as per Watson and Crick model, the DNA which was described was the B form. So now it is known that DNA can exist in several other forms. And the primary differences between the forms is in the direction in which the helix spiral, how the helix is spiraling, whether it's on the right hand side or whether it's from the left hand side. So A, B, and C, these type of DNA, they have got right-handed helix. And Z DNA, it has got a left-handed helix. But this doesn't occur naturally. It is found in vitro. You can make a Z DNA helix or under high salt concentrations. So the B form is the major form that is found in the cell. Z-DNA was initially found under high salt concentration, but the cellular environment is actually a low salt environment. So the question is whether type Z exists under cellular conditions. So several features have been discovered that can stabilize Z-DNA molecule under low salt environment. So you had found that Z-DNA, it exists under high salt concentration, but this there are several ways by which you can do it with low salt environment. So what are the cases where you have alternating purine and pyrimidine tracts? No more students will be allowed anymore. So whenever there is poly GC or poly AT stretches, then there is GC or AT long stretches, then this Z-DNA structure can exist. And when there is a 5' methyl cytosine added to the DNA. So these conditions can exist in the cell, but actually they are existing in between stretches of bDNA. You have lots of bDNA, then suddenly you have a little bit of zDNA, then again a bDNA stretch. It works like this. In addition to the direction the molecule turns, several other differences exist between the various forms of DNA. So what are the differences? First, whether it's A, B, C, or Z. Then the direction of the turn, right-handed, turns from the right, or whether it's left-handed. And number of bases per 360-degree turn. What are the number of bases per 360-degree turn? 11 bases in A DNA. And the diameter of the helix, 23 angstrom. B DNA, you have known 19 angstrom to 20 angstrom. And you have 10 bases per 360-degree turn. And in cDNA, it is right-handed helix, and you have 9.3 bases per 360-degree turn. And the helix diameter is 19 angstroms. Now you have the famous zDNA, which is left. Instead of 10, you have 12. And it has got an 18 angstrom difference. So you can see that there are certain differences present. So this actually concludes the different types of DNA. Next, we will go to the next part. This study material has been given in the Google Classroom about physical properties of DNA. So you have the first thing is hyperchromic shift. So you know that DNA molecules, here it's increasing temperature, and it is here is increasing absorbance. So light, UV light at 260 nanometer is passed through the DNA molecule, and then uh, depending on the concentration of DNA, more amount of UV is absorbed. So this absorbance, so more the absorbance, more the concentration of DNA. So in this hyperchromic shift, what happens that the phenomenon of UV absorbance, it as DNA is denatured. And this phenomenon is known as the hyperchromic shift. The purine and pyrimidine bases in DNA, they strongly absorb UV light, and double-stranded DNA absorbs less strongly than denatured DNA due to the stacking interaction between the bases. So when the molecule is DNA molecule is something like this. So there is this stacking one upon other. And so in that case, this absorbance is actually lower. So you can see this double-stranded DNA, it is giving a lower absorbance. And then the temperature increases, the DNA melts down. And gradually the double-stranded is becoming single-stranded DNA. The helix is opening. And here the absorbance increases. And when both of them have become single-stranded, you have the highest absorbance. This is known as hyperchromic shift. Now, we deal with when there is repetitive sequences. When a particular sequence, DNA sequence, DNA sequence repeats several times. When a DNA sequence, then there there are some different changes. So in a whole genome, if you want to understand if this particular genome or DNA sequences has got lots of repetitive sequences, you can understand it. So first, these are the two DNA strands. These are the two types of DNA strands. So what happens when you mix these two strands, repeated strands, then you denature them at high temperature or high alkali, then they become single stranded. And gradually you lower the temperature or remove the DNA, they reassociate. And then the reassociation can be either you have this strand fully, or it can be something like this, or it can be hybrid, or it can be a total something like this of both the strands. And so we see that a substantial portion of the eukaryotic genome, it consists of highly repetitive non-coding DNA sequences. The coding sequences which code for proteins are very low, but in the non-coding DNA sequences, what happens is that they have highly repeated genomes, repeated sequences. They repeat several times. Sometimes they are present in hundreds and thousands of copy per genome. The same non-coding region, it is repeated hundreds and thousands of times. They were first demonstrated by Roy Britton and David Cohn during studies of the rates of reassociation of denatured fragments of cellular DNA. So you denature the whole genome, make them single stranded, and then you allow them to reassociate. So if there is highly repetitive sequence, similar complementary system, they will join faster. And if it is unique sequences, the repetitive sequences are not there, then they will join slower. More repetitive sequence, the DNA joins. More repetitive DNA, the sequence, the DNA joins faster. And less repetitive DNA, the DNA reassociates slower. Now, on the basis, it is a bimolecular reaction. This reassociation is a bimolecular reaction. Two separate strands of denatured DNA must collide with each other in order to hybridize. पहले खोले हो, फिर then you are reassociating it. So there is a two ways bimolecular. You need two molecules to collide, and the rate of reassociation depends on the concentration of DNA strands. So it was seen that when fragments of E. coli DNA were denatured and allowed to hybridize with each other, all the DNA reassociated at the same rate as expected if each DNA sequences were represented once per genome. So in E. coli, repetitive sequences are not there. So all of the fragments, they reassociated at the same time. But in the mammalians showed a very different result. What he found that approximately 60% of the DNA fragments reassociated at the rate expected for sequences present once per genome. So 60% of them, they were non-representative. They are present once in the genome, non-repetitive. They are present once per genome. But the remainder reassociated much more rapidly than expected. So, and this actually is the repetitive. So, interpretation of this result was some sequences were present in multiple copies and therefore they reassociated more rapidly than those sequences that were represented only once per genome. So what they use for such a study is a analysis known as COT study. So CO is the initial concentration. So this kinetics COT value. CO is the initial concentration of DNA. Initial concentration of DNA multiplied by the time of incubation. Initial XYZ मोलर मल्टीप्लाइड बाय द टाइम 10 मिनट हम लोग ने इंक्यूबेट किया था टाइम ऑफ इंक्यूबेशन so for it to reassociate so you can find that the DNA E. coli DNA reassociates at a uniform rate but in the mammalian something like 60% of the genome the non-repetitive sequence they reassociate more slowly than E. coli DNA. So this is how the COT curve looks like. This is the COT value over here and genome reassociation and this is measured by absorbance. Single stranded, double stranded, single stranded more DNA, more absorbance, double stranded less absorbance. So यहाँ पे क्या किया the COT value gradually all of them have more the time it goes and initial concentration of DNA the absorbance goes down. But here in the calf it goes gradually down. So this pink region, simple sequence DNA, non-repetitive, repetitive. This brown color, it is the intermediate repeated sequence over here. And this much, 60% of the genome, this is single copy DNA. So initially single copy बढ़ गया यहाँ पे डबल कॉपी आ गया सेम रेट में हो गया लेकिन यहाँ पे सेम रेट नहीं हुआ है almost the same rate. So this is COT values. So now let us discuss this in details. COT analysis, it is a technique based on the principles of DNA reassociation kinetics. It is a biochemical technique that measures how much repetitive DNA is in a DNA sample. So what is the COT value? The cot value is the product of C0, the initial concentration of DNA. यहाँ पे measuring the initial concentration of DNA in this region, multiplied by the time and a constant that depends on the concentration of the cations on the buffer. So this you need not multiply, take into account. So moles into second, mole second, molar seconds. A repetitive DNA will renature at low cot values. So रिपीटेटिव होगा तो बहुत फास्ट रीनेचर किया, देखो ये बहुत फास्ट रीनेचर किया, वैल्यू कम्स डाउन, while unique DNA sequence will at higher cot values. So this is cot analysis. The next thing, which is TM. How do you define the TM? It is the melting temperature. It is the temperature at which one half of the DNA duplex will dissociate to become single-stranded based on the base pairs in DNA 1 and DNA 2. So the temperature at which 50% will become 50% are single-stranded, 50% are double-stranded. This is TM values. The next thing which we will go is the C value paradox. Now, what is the C value paradox? It's also known as the C value enigma. So the C value represents the amount of DNA in the haploid genome of the organism. The C value, it represents the amount of DNA in the haploid genome of an organism. So haploid genome में कितना amount रहता है? So that is the C value. So, and what is the genome sizes vary and enormously among species. So between different species, you have different amounts of DNA. And more complex species, it is expected more complex species will have more C values, more DNA. Less complex, less C value. However, this is not found throughout the genome. And this is very puzzling. So more complex matlab hum log sochte hai, we think that there is more number of genes, but this is not true in this case. And therefore it is a complex puzzle. So genome size does not correlate with an organism's complexity. So it's a challenge about DNA content and evolution. So you cannot draw a parallelism. So, for example, salamanders, they have 40 times more DNA compared to humans, even though humans are more complex organisms than salamander. इसका कोई correlation नहीं है, किसी का ज्यादा है, किसी का कम है. And so this ends also C value paradox. So do you people have any questions? कोई questions है? Hello?
No, sir.
Okay. Then
No, sir.
Tata bye bye today.
Thank you, sir.
Thank you, sir.
You're welcome.